Hello.
Hey, how are you?
I'm good to a certain extent. I'm good. I can't complain. I'm okay. I'm okay.
What's new?
Um, so I definitely like went a little crazy last week where I was kind of just like studying like all night and
Hmm.
every day, and I wasn't sleeping as much as I should have, and um kind of like spun out. No, I didn't spin out crazy,
Hmm.
but like I took my test yesterday and I was just so anxious during it. Like I feel like I didn't do as well as I could have in the Chem Phys section. Like, I mean, I just got like a 500, which I was kind of upset about just because I was hoping to see like, that improvement because I was working so hard. But I think like
Yeah.
I wasn't in a good frame of mind to take the test, which is
Hmm.
okay. I'm happy like at least like I got that like I don't know, pre-exam jitters like out the way before it's my actual test, but I'm just like, I don't know.
Got it. Yeah, so for for this week we could kind of pretty much take it easy for the most part. Um and you took this test, when was it?
Yesterday.
Okay. And what was your breakdown? I know it's like lower than what you wanted, but
It was a 124 in Chem Phys, a 124 in cars, and
Mhm.
then 126 in Biochem and a 126 in Psych Soc.
Okay. So you're still, 500 and up. So I think that it was mostly just like staying up and, not getting enough sleep and stuff like that. So we could just, maybe go over some of it, but for the most part this week we could kind of take it easy. Focus on making sure you get enough rest, get enough sleep, um, all of that stuff.
Yeah.
Mhm.
I'm probably gonna like take some sick days for work just so I have like enough time to like just prioritize this. I mean, because like right now I'm over here like still typing at work just because I don't know, just working. I feel like it's crazy, like working on a research project that involves people. I feel like is so like
Mhm.
involved. It's just very like I feel like I'm very involved like mentally with that, so it's hard to like turn
Yeah.
it off and like separate myself from that.
Mhm.
So it's fine. Like
Yeah.
I'm like y'all will see me next week because even for me to like tell them that I'm probably gonna take these sick days, I feel bad, but it's fine.
Yeah, you got to do what you got to do.
Yeah. And they will be there next week. I don't know.
Yeah. All right.
Yeah. Okay, yes. Let me stop. No more work. No more, no more, no more. Okay.
Mhm.
After 5 anyway.
Mhm.
So I redid the review for Chem Phys.
Mhm.
It's like, oh, some of the questions that I got wrong, I was definitely just like, I don't know. Like I felt like I knew how to do it.
Yeah, yeah.
But also some of the questions that I did get wrong, I was like I feel like some of the OChem questions, I think I need to work on that, like figuring out like what
Hmm.
reactions. Actually, I'm gonna go this way. Like the reactions I need. This is fun. So I redid these. Um, just something like this. This question I got it wrong because I was thinking, oh, I feel like this is too easy of an answer. So I was like, let me go with something that it seems a little bit more complicated, but it was actually this. And I was just like kind of annoyed because I'm like, oh, I overthought it.
Hmm.
Um, this I just didn't know like the tertiary alcohol to ketone.
Hmm. Yeah, so um in like the PDFs folder, I have like a generalized biochem redox pathway like PDF, and in it you can see like in general just what happens when you have redox, like for like organic molecules. So basically, so let's say we have an alkane like this. So you could basically have three things happen. You could either have loss of hydrogens, you could have gain of oxygens, or you could have like gain of one oxygen, or you could have gain of additional bonds to already existing oxygens. So if we oxidize this, first thing that'll happen is it'll lose hydrogens. So this will just be, oops, an alkene, like a double bond. And if you oxidize this, since we've already lost hydrogens, now we can just gain like an oxygen. So we can gain an OH group like this. So this will give you an alcohol, and you can have your, so if you have this is a tertiary alcohol, and then this would be a secondary, and then, oops, and then this would be primary. So now if we oxidize this further, so we've already lost hydrogens, we've already gained an oxygen. So now we're going to gain an additional bond to the already existing oxygen. So for the secondary, you can do that, right? You can
Hmm.
add this. What is this molecule?
That's a ketone.
Good. And this, you can get this, right?
Yeah,
That's a what?
the aldehyde.
Okay. Now for the tertiary one, there's no more, like this carbon is attached to three other carbons and the OH, so it can't be oxidized any further. So this stops, like once it gets to the tertiary alcohol. Um, the secondary alcohol could be oxidized into this ketone, but it cannot be oxidized further.
Hmm.
Stops there. Now the aldehyde here can actually be oxidized further. You can add, so we've already added like an additional bond to an existing oxygen. So now we can add an additional oxygen to get
Yeah.
a carboxylic acid. And then this could be oxidized again. So now we have like one oxygen with two bonds, and the other oxygen could now have an additional bond to get carbon dioxide. Let me make it more right here for CO2. And that's the last, you know, thing that something can be like a organic molecule, a hydrocarbon can be oxidized to, which is why you see it at the end of like the electron transport chain with,
Okay.
Mhm. Like when the electrons eventually go to oxygen, all of the carbons eventually leave as CO2.
Okay, so
Hmm.
the reduction potential, like at, well, I guess it's like oxidizing, so the reduction potential as it's getting oxidized is decreasing.
Oh yeah, yeah. Um, so if we're talking about reduction potentials, whichever, and we're talking about like in a redox reaction, right? whichever has the higher reduction potential is going to be the thing that gets reduced, and whichever has the lower reduction potential will actually get oxidized, which means that um, whatever value you have for the reduction potential, you just flip the sign. So like if you had, let's say you had something with a reduction potential of 1.17 and then something with a reduction potential of 1.6, which one will be reduced?
Um, the one with the 1.6.
Hmm. So that means that this one will have to be oxidized. So then this reduction potential value no longer is really relevant because it's going to get oxidized. And its oxidation potential is going to be exactly what its reduction potential is, but multiplied by -1. So its oxidation potential will be -1.17.
Okay.
Yeah, so that's just like, yeah, reduction potentials and oxidation potentials. Um, but up here, this is, well, yeah, in this direction, it's oxidation. In this direction, it's reduction. But I basically have like, yeah, a PDF like this in the PDFs folder that you can like pretty much like memorize. Um, because uh like there's a lot of ways they can ask redox questions, and usually like the definitions that we have, like loss of electrons is oxidation, gain of electrons is reduction. Like that's mostly useful for like in Gen Chem when you have like metals that change
Mhm.
oxidation states, but for like organic chem and biochem, you can't really see changes in oxidation states. You instead see like, yeah, like either if it's oxidation, like loss of hydrogens, gain of oxygens, gain of bonds to oxygen, and reduction will be in the opposite direction. So here you're asking which of these are unlikely to have to occur, like oxidative reactions. So a primary alcohol to an aldehyde, that can happen. Aldehyde to a carboxylic acid can happen. Secondary alcohol to a ketone can happen, but tertiary cannot be cannot be oxidized.
Gotcha. And then this I just didn't know what the difference between Z and E is.
Okay. So uh so Z um means, well, like Zusammen, but it basically means that you have things on, pretty sure that's when you have it on the same side. Yeah, together in German. And E is entgegen, which is when they are um on opposite sides, meaning like the relative to the double bond, and whatever has the higher um priority will be like the bigger part. So for instance, if this is our double bond, let's consider like this like the line of symmetry. And so on the left, the higher so there's an hydrogen here. So the higher priority is this methyl group, right?
Compared to the hydrogen, yeah. Oh wait, so it's talking about like the chiral center?
Um, n
Wait, no, never mind. Never mind. Sorry, I got confused.
mhm. Yeah, just basically relative, like it's basically another way of saying like cis and trans.
Oh, okay. Yeah.
So Z is basically cis and E is trans. And so and you go by whatever has the higher priority, aka, the larger like substituent. So here on the left, this over here is the higher priority one. And then this on the right, even though there's a methyl up here, there's an ethyl here, right? So the higher priority one on the right is this guy. So this is going to be so both on the same side of like the double bond. So it'll be like Z. So we can get rid of these two. And then we just finish the naming part. So the overall molecule we know is gonna be like ene because of the double bond. Um, and then this part is the longest chain. And so I'll use red. That'll be 1, 2, 3, 4, 5. So the longest chain is 5 carbons long. So it'll be the pent part. And then they're saying that at the 3 position, which is over here. So actually this is a good, I guess, thing to look at. So because you could have the ethyl part here, and then the longest chain could be, 1, 2, 3, and 4 to get ethyl butene, but then that won't be the longest chain anymore.
Yeah.
The longest chain is the 5 carbon one, which is pent, and then that means that this 3, uh the substituent at the 3 at the third carbon is the methyl.
Okay. Gotcha. This like I think I just need to like go over the units and memorize like the different ways that they could be transferred because like when I was going over this like I realized that Hertz is 1 over S. So I gotta get that one in my book as well.
Yeah.
Um
Mhm.
the is okay. I was like kind of confused by this one, but I mean,
Okay.
so
So maybe you chose A.
yeah, I did, yeah.
Yeah. So from this figure 2, we certainly see that it seems to perform the best when you have both of them.
Mm-hmm.
So then, then it's, I guess, the question of like, do you want to administer both in like at the same time or one before another? So, um, okay, so let's see. So they're saying that assume that the cellular uptake rates and drug delivery rates of compound 1 and 2 are identical. Um, if the proposed mechanism of sensitization by compound 1 is correct. So they're talking about something with compound 1 only. doing something called sensitization. So, that means like let's investigate, like let's look at what that's about. So, let's see. Can you go up on the left? Okay, let's see. So, chemotherapies initiate apoptosis, and this is a protein, NFKB, okay. And then they talk about the imidazoline family inhibits this NFKB pathway. Based on this observation, researchers were interested in determining if a member of this family of compounds, compound 1, figure 1, could selectively sensitize cancer cells to the potent chemotherapeutic agent camptothecin, compound 2, figure 1. Um, Compound 1 was chosen from a small library of similar compounds based on its ability to inhibit degradation of IKB. Um, all right, so they're talking about compound 1 doing something like whatever this means, right? Sensitize cancer to
Hmm.
the chemotherapeutic agent compound 2. So, so that they talked up here about how chemotherapies um, can initiate apoptosis, and that could be like a way for them to kill cancer cells. So we know that when they describe compound 2 as a chemotherapeutic agent, we know that compound 2, like that's what it does. It can start apoptosis, and through that mechanism it can kill cancer cells. But for compound 1, they didn't say it was a chemotherapeutic agent, right? They just
Yeah.
that it could sensitize cancer cells to the actual chemotherapeutic agent. So maybe, so like, we have to, I guess, like think about, okay, what does that mean to sensitize something to something else? And I mean, like, I could say, like, yeah, it probably means makes it more sensitive to the um chemotherapeutic agent. But let me see if there's anything in the figure to help us out. Um, okay, so when we have compound 1 only, right, over here, it's as if
Yeah.
it's the same results as if you have no drug, right? So,
Hmm.
so like, so yeah, it doesn't seem like compound 1 is a chemotherapeutic agent. And when we look at if you administer compound 2 only, it goes up, right? It's
Yeah.
not like immediate, right? But it certainly goes up a measurable amount. And so now, like, I want you to have like, more than one kind of way to figure this out. Because like on one hand, you can see them saying sensitize and see that to mean that it can make it make it more likely to be affected by compound 2. But I also want like because like they could say all sorts of stuff and it could mean all sorts of stuff to be honest. So I also want to give you the ability to look at the at the figure here and see that okay, like from I guess the paragraph above, like they were saying that compound 1 was something and compound 2 was something else. So we know that they're different. And now here we can see, hey, compound 1 doesn't really do anything on its own, and compound 2 on its own does do something. So, and then if the combination of the two has the best results, then maybe what they mean by like sensitize is that it can get the cancer cells ready for the chemotherapeutic compound to kill it. So, so we have simultaneously, we have compound two first, and they only differ by, some time. And then we have compound one first, and and yeah, like, I totally understand, seeing this and thinking this, but the rest of the passage um tells you that compound one sensitizes it, which would mean that it would like kind of get it ready for it to be affected by compound two.
Gotcha. Yeah, I see now. Ugh,
Hmm.
I don't know if I was moving too quick. Oh, just I missed it, I guess. Um, this question, I thought it was sodium, just because I was thinking about electronegativity, and I'm like, I'm not. My bad. No, no, no.
Sodium turns into what type of ion?
Positive.
Which means,
It loses
uh-huh.
electrons. Wait, hold on. Oh, okay, that just.
Hmm.
Okay, so then I guess it makes sense that it's this
Hmm.
one then.
Yep.
I was confused. I was between these two, and I thought yeah, I figured this was more electronegative, which is why I was like, well, would it want
Hmm.
any more electrons?
Yeah. So that like electronegative means it really wants electrons.
Okay, yeah,
And then, but yeah, you
gotcha.
think about what type of ion it'll make.
I think I just need to memorize this phase diagram a little bit better. I mean, like when I did it the second time, I realized it was a critical point, but I just need to memorize where everything is.
Oh, well, so like yeah, this will depend based on what you're looking at, but
Oh,
well, I mean like um I just mean that in the case of water, it looks a little different because water as a solid is less dense. But for most things it'll look like this, but like for water it'll look more like that. But yes, your like in general, your whatever the point is, like okay, so we could see like so for instance, point A separates CO2 gas from CO2 solid. So point A has to be like the point at which a solid can turn into a gas or a gas can turn into a solid. So that's going to be deposition if it's gas to solid and sublimation. So yeah, that'll be that. And then melting point would be between the solid and the liquid. So probably point C here. No, 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 that's between liquid and gas. Melting point would be, well, not here, but somewhere on this. And yeah, the critical, so now point B here is called the triple point
oh, okay.
when, because it's like at the interface of the three phases. Um, but then point C here, yeah, it's called the critical point because beyond that point, there isn't really a difference between like the liquid and gas. Kind of exists as both. It's kind of weird.
Oh, so it's not the critical point at A?
Sorry. So the trip, wait. Oh, wait, no. Wouldn't that be the sublimation?
Oh, I thought it was like, okay, wait, I don't know. Wait, sublimation solid to gas. I guess, yeah.
Mm-hmm. It could also be called like the deposition point because, but yeah, you're essentially going to look at, so for instance, like A, where is it? It's on the line between solid and gas. So point A is going to be named, something that could be like solid to gas or gas to solid. So solid to gas, like, without going through the liquid phase, like straight up solid to gas is called sublimation, and gas to solid is called deposition.
Okay.
Yeah.
Um, Actually, I should got this one right. I messed that up. Um, so for this one, I thought it was covalent bonds because it says here that RT in its binding pocket has the valine and the phenylalanine and
Yep. So what type of amino acids are these?
uh nonpolar.
Mhm. So they would be hydrophobic, right?
Yeah, that's that. Like, I got it like the second time around, but
Mm-hmm.
I guess I'm just kind of confused because I'm like, how is it supposed to? It's not necessarily bound. How is like the how is compound two bound to the site then in the binding pocket if it's just a hydrophobic like interaction, I guess? Like, wouldn't it
So,
just like push it away?
uh, so, um, let's see. So your covalent bonds are going to be like the really strong bonds that you draw like with the lines, like, each of these lines is a covalent bond, and that's like
Mm-hmm.
really strong. But typically when you're dealing with enzymes, right? Um, your enzyme will have some type of active site where a substrate can go and form, temporary bonds with, and by temporary I mean non-covalent, like intermolecular bonds, like hydrogen bonds or hydrophobic interactions, those types of stuff. Um, and then the enzyme can turn the substrate into a product and then it can leave. Um, and then here they're talking about an inhibitor. So an inhibitor can bind to like a non-active site to cause like an allosteric effect. Um, but this one is binding to the actual binding site. So this is a competitive inhibitor. But but yeah, you typically have when you have enzymes, uh well, even like in a proteins, so like in a protein, like only the primary structure, um, of like the peptide bonds that are amide bonds, that's a covalent bond. But then when you go to like secondary structure, you have hydrogen bonding between the backbone, right? Um, Tertiary structure, you have hydrogen bonding between the R groups, you have electrostatic interactions, you have um, dipole interactions, you have van der Waals, but mostly hydrophobic interactions. There is one covalent bond that can play a role in proteins like, tertiary structure, and that's like the disulfide bonds. Um, but but yeah, that's the only one. And in addition to the primary structures, peptide bonds. So most of the bonds are going to be from like something like these. Like hydrogen bonding will be like kind of like the strongest type of intermolecular bond. Um, But yeah, it'll be mostly that. So we can see that these compounds are non-polar, right? Like they're
Yeah.
rings and benzenes and stuff like that. I mean, like, there's amines attached to it, which could participate in hydrogen bonding. There's stuff in here that could participate in hydrogen bonding, but even that would just be a still an intramolecular um, bond, right? So, so typically you're gonna see like, passages like this a lot. So typically, so like here, like big picture. So like this is reverse transcriptase, ends in ASE, so it's an enzyme. And then they tell us, okay, it's a heterodimer, so it's composed of two subunits which are different. Um, and they tell us the weight of one, the weight of the other one, and that we can have RT inhibitors. So they say RT is the name of the enzyme, so we could inhibit the enzyme using these compounds, right? And so we take a look at the compounds and they're all, hydrophobic. And then they typically will tell us about the active site of the enzyme. So they're telling us that the active site, let's see. So they're saying compounds 1, 2, 3 through 3 bind to a site that's not the active site, and that the binding can occur at any stage of substrate binding. So, okay. So it's not binding to the active site. So never mind about what I said about it being a competitive inhibitor, but it does bind at some point. So it says that it binds. So the inhibitor binding pocket, aka the part on the enzyme that the inhibitor, one of these, binds to, has valine and phenylalanine. And usually that would kind of give away what type of interaction is going to be predominant there. Like if they said serine and threonine, we would likely see like a hydrogen bond. There.
Hmm.
If they said like glutamate or like lysine, it'll be some kind of like ionic bond, or interaction, or ionic like that. Um, And then, yes, if they're saying valine and phenylalanine, um, those are the hydrophobic, aka nonpolar amino acids. And we already see that these inhibitors, like they're asking specifically about compound 2, but for the most part, compound 1, 2, and 3 are all nonpolar, therefore hydrophobic. And so it'll make sense that they can bind to this pocket that contains valine and phenylalanine. Right.
Okay.
Hmm.
Sounds good. Um, this was just like I guess like the math equation, but I feel like I don't know, I don't think I did it right. It says I think I was just off by like a couple of zeros.
Okay.
So it gives us the equation here where it's like the absorbance is equal to this
Hmm.
E times B, which is the length of the sample cell, and then times C, which is the concentration.
Hmm.
So it's asking about the concentration. So I set the equation equal to zero point 0.7. Well, zero point 0.7 equals um this.
So let's see. Absorbance is equal to this times this times this. So we can divide both sides by this. So that is 1367. And then B was 1 centimeter,
Hmm.
but this has 1 centimeter, like, as a denominator.
Yeah.
That kind of just goes away. So then I would just do 7 over 10. Oops. 7 over 10 times, I guess, 1 over. 1400 goes into this, 200 times 1 over 2000, right? So if
Yeah.
1 over 2 is 0.5, this is going to be 1, 2, 3 of these guys, which is just 5 wait, 1 over 2 is 0.5, 1 over 220 is 0.05, 1 over 200, 0.005, 1 over 2000. Okay, so that's that, which is okay.
Right.
Yep.
Okay. That. Um, -4 micromolar.
Oh, that's in wait.
Wait, my bad. Sorry. Confused.
7 over this times 1 over this. 7 goes into this 2 times 200. 1 over 2000. Okay. Let's see. 500 micromolar would just be oh. Oh, okay. Because this would be 0.5, oops, micromolar, right? 0.5 micromolar. I was thinking 5 micromolar.
Okay, wait.
Mm-hmm.
Um, so times 10 to the -6, 0.5 times 10 to the -6 would be this here.
500 times 10 to the -6, right? So he did 500. 1, 2, 3, 4, 5, 6. So that's 3 zeros and a 5.
Okay, okay, yeah, I see.
Yeah.
Okay. I thought you were saying it was zero point 0.5 micromolar. Oh, you were saying zero point 0.5 millimolar, my bad.
Mhm.
Okay. Ah, and then this time I got this one. I don't know.
Yep.
I think I was just tweaked and I bugged a little bit.
Sure.
Um, okay, this one I thought that the answer would be the catalytic efficiency decreases with pH and the velocity. I just didn't think it would be the same. I didn't think I just thought the velocity would increase. I didn't think there would be the same relationship between them.
Increasing pH would have what effect? It seems like, okay, so this is the catalytic efficiency, right?
Yeah.
And then this is the catalytic turnover rate, but this is equal to Vmax over enzyme concentration. And enzyme concentration
Okay.
like a constant value. So in other words, kcat is proportional to Vmax. So if kcat goes up, Vmax goes up. Okay, so it seems like kcat goes down, so Vmax goes down, and then catalytic efficiency goes down. Um, so yeah, both of them.
Okay. Okay, good to know that KCAT is related to it's
Mm-hmm.
proportional to Vmax.
Mm-hmm.
Um, I think I just need to go over these terms. Um, I thought it was refraction, but it's diffraction. Um, I'll look over that.
Yeah.
And this, I was just looking at, I looked at this completely wrong. Like I was looking at the figures wrong.
Yeah.
And got this. Hmm. I was a little confused by this. It says here, how many hydrogen bonds are present in the two types of GT mismatches? Um, so for this, does this mean like instead of the GT, the A binding to the T, it's the G binding to the T?
Let me see.
Because like I didn't have too much time like at the end of this
Hmm.
exam, so I definitely like was rushing towards like at this point I was rushing bad. I normally always have time. That's how I knew I was like
Hmm.
I don't know what I was doing.
Um, let's see. They're saying that DGTP could be added instead of DATP. GT mismatches occur in two different ways. In the wobble base pair, the GT mismatch appears as an offset AT base pair. In addition, GT mismatch can mimic a GC base pair if the enol tautomer of T is present. And they give us the tautomers, keto form, enol form. Um, okay. How many hydrogen bonds are formed and the two types of the mismatches.
So I figured like
Mhm.
if so it would be a G to T instead of an A.
Yeah.
So and if like because G is the purine.
Mm-hmm.
Yeah, G is the purine, and I feel like don't they have three hydrogen bonds?
Yeah, G and C could make three hydrogen bonds.
Yeah, so I figured like it would be three in the wobble pair and three. I thought it would be three and three.
Offset AT base pair. How many hydrogen bonds? So we know that the enol form gets rid of the keto part. And I mean, I guess like okay, so this could accept a hydrogen bond, but this can donate a hydrogen bond. Same thing here. Um, how many hydrogen bonds are present in the two types of the mismatches? Polymers and GMC, and then we're saying T here, and then terminal. base pair. GT mismatch appears as an offset AT base pair. Okay, so if it's wobble, G turns to A. So I know GC will have 3 hydrogen bonds. But if this is wobble, it's no longer GT, but it's AT. So my guess would be 2 in the wobble. And then in addition, the GT mismatch can mimic a GC base pair. Now that is 3 hydrogen bonds. GC,
Hmm.
if the
Oh,
E
okay.
tautomer is present. Yeah, maybe that one.
Nice. Okay. Mm.
Yeah, that was kind of tough.
It's just confusing because it says in the wobble base pair.
Mm-hmm.
I thought I thought that the GT was the wobble base pair,
Mm-hmm.
but I guess I don't know. I guess it's just like wording. I don't know. I was tripped up.
Mm-hmm.
Um, this I could go over this one. I could do that one myself. This like I was so annoyed by getting this question wrong because I picked the right answer and then I changed it like at the last minute. Like I don't know why I did that. Um, and it's diastolic over systolic, right?
I think it's systole over diastole
Oh,
because
okay.
I think systole is the um when the left ventricle when the ventricles contract and when the left ventricle contracts, it has to push that blood through the rest of your body. Whereas if the atria contract, it just pushes it into the ventricle, right?
Hmm.
So it's like atria contracts, the ventricle fills, and then the ventricle contracts. The atria can fill, but it also push like all that blood through the rest of your body.
Okay. This kind of pissed me off. Um, just because I'm like, well, it's like what occurs first during action potential generation, and I figured like, oh, you have to reach the threshold potential first.
Hmm.
But I guess like I wasn't putting two and two together, where it's like, because I'm over here thinking like, oh, once you reach threshold potential, then like all the sodium channels will open up and it'll generate the action potential.
Hmm.
So I was thinking like. I don't know what I was thinking, but basically, I guess the answer is that the um sodium channel is open at the hillock.
So let's say that, you're thinking like the way that you should, but there doesn't seem like there's any like choices that would be like, one that would say like the threshold has to be reached, right?
Hmm.
Like we have this, which is just, the potential, like the action potential has already happened. It just, travels through the axon. This only happens at the neuromuscular junction, but it would happen after um the action potential. And then hyperpolarization is, when it goes back down too much. And then this would be
Yeah.
the first thing. So yeah, like you're right to think, you must reach the threshold potential. Um, but then when you look at the choices, none of them seem to say that, right?
Yeah, I think I just clicked like graded potentials propagate along the axon because I was like, oh well, they're not gonna catch me because I'm like, oh the voltage gated sodium channels don't open until it's like actually happening, but
But
I
also,
still.
if we think about where these things are, right? Like So like this entire long part's the axon, right?
Hmm.
Like this is like the soma or like the cell body. And then this part here is the axon hillock. It's a weird word, to be honest, but um but yeah, that's there. So you can think like if you were, thinking about this one, you can think, all right, well, this is all right. This is past the hillock.
Oh, okay, yeah,
That could help as well.
good to know. Oh, MCAT, and then yeah, that's it for this section. Um, I started doing the BB. I didn't get too many of these wrong though.
Hmm.
I'm doing better in biochem. My goal is to like only get 10 questions wrong on test day. Like I still feel like I could do that. I think I just need to do more practice questions.
Hmm.
Oh, yes. I realized that like I just didn't know what cytochrome, like what the cytochrome P enzymes do. Like I kind
Yeah.
of just guessed.
Yeah, this is kind of annoying. Um, because like nothing really, I mean, a lot of things here talk about phosphorylation, so they might make you think that, but yeah, I guess if you can think of any cytochrome, like cytochrome C could come to mind, and that's, in the electron transport chain and all that stuff is like redox. But yeah, this could be annoying.
Yeah, so I guess it's oxidizing them, right?
Hmm.
Yeah. I'm like, okay, I know that one now. Ooh, my God. And then I need to go over fatty acid synthesis because, um, yeah, I feel like a lot of the questions that like I've been getting wrong in this section are like questions that like the pseudo discrete ones where it's like tied to the passage, but like you have to know like, what happens during the fatty acid synthesis. So this is
Hmm.
yeah, what functional group is added? It's this one. I thought it was a OH for some reason. I don't know. Oh, yeah, I know. Okay. I need to look at this and figure out what Henry's law is. I mean, I think I was thinking about Charles's law, because I put the volume, but that was wrong. Um, so yeah, I'm gonna spend some time with my equation sheet this week and just really make sure I memorize like everything. Um, and then this tripped me up because it said there was 28.
So I'll just say for Henry's law, like basically it just means like if you have a higher partial pressure of a gas above a liquid, you can think it's a higher partial pressure, so it's gonna push down um against the liquid and dissolve into the liquid. It's so like if you're like diving, um like going underwater and stuff, you're going under larger pressure. So your blood, so like the gas, like air, the partial pressure, well, air is above your blood, and the pressure of air is large, like increases, so air gets more um diffused into your blood. But like they're saying partial pressure, which means like for a specific like gas. Like I just said air, but air contains oxygen and nitrogen. So
Mhm.
those things will go like the partial pressure of those things will go up as well. Um, and like the bends, have you ever heard of that? If
Yeah.
Yeah, it's like if you so let's say you're already like at a depth, but you really like you ascend really quickly, you go up really quickly.
Hmm.
Uh, the drop in that pressure makes it so that the dissolved oxygen in your blood can, turn back into like gaseous oxygen. And but the same thing with nitrogen. And when nitrogen like bubbles up too much, then you get something called the bends. But yeah.
Gotcha. Um, for this one, it says here that it's a 28 amino acid peptide hormone. So I figured I would just do 28 times 3 and that it would be 84. But apparently, you have to factor in the stop codons. So it would be more than 84. Well, more than 87 nucleotides. But I was kind of confused because I'm like, well, if you have to factor in the stop codon, like why won't you factor in the start codon?
So the start codon actually still codes for an amino acid, methionine.
Hmm.
Yeah.
Okay, so it's like the stop codons are part of the protein.
Um so so the start codon like will still have a methionine like attached to like the tRNA. So the start codon will have an amino acid associated with it. So basically when a protein starts getting translated, um it'll start with that methionine. But
Hmm.
the stop codons don't code for any amino acids, so that's just when it just leaves the ribosome.
Okay, so they're just like hanging out, I guess.
Yeah. So basically, start codon starts translation and codes for methionine, an amino acid, and every other codon will code for an amino acid, but your stop codons won't code for any amino acids. They'll just stop the um, peptide from being made.
Okay. Okay.
Mhm.
Um, this one also kind of had me a little tripped up because it says here hormone replacement therapy is often given to children who have the Prader-Willi syndrome once they have reached the hyperphagic stage. So I figured that the therapy would be to prevent like the excessive eating that happens with hyperphagia. Um, but I don't know. Apparently, that's not. I was confused because I wasn't quite sure if it was giantism because I thought giantism was like that was when you like can't necessarily stop growing and you get like super duper tall. So I didn't think it would be that, but I don't know. I figured that none of the other answer choices were making too much sense to me.
So they're
But
about hormone replacement therapy with growth hormone, right?
yes,
And then like if you start that, what would be prevented?
yeah.
So what could be prevented if you're given growth hormone?
Well, I guess like probably being like short.
Mhm.
Okay, but what does that have to do with the hyperphagic stage?
They're just saying like this is I guess some disease that let's see. So at first, seems like they have feeding difficulties, but then later they have an insatiable appetite, like they just eat a lot. So I guess like if they said before the hyperphagic stage, then you know the growth hormone could lead to them eating more.
Oh, okay. I think that makes sense.
Hmm.
Okay. Okay. This passage was hard for me. I was confused.
Hmm.
Um, okay. Should I read this?
Hmm.
Okay. Muscle cells are equipped with inherent capacity to perceive and respond to biomechanical stimuli, such as pressure, and convert them into molecular events that regulate multiple anabolic and catabolic processes. Myod1 is a member of the family of helix loop helix myogenic factors that play a major role in regulating muscle differentiation by binding to the consensus sequences of the regulatory region of muscle-specific genes such as cyclin-dependent kinase inhibitor 1A. I highlight him. Myode-mediated induction of CDKN1A results in the irreversible exit of undifferentiated cells from the cell cycle and their commitment to program, different program. Myode contains a C-terminal nuclear localization sequence that promotes its translocation to the nucleus.
All right,
Um,
let's do some proportionality stuff. So Myode 1 activates this guy, right?
mhm,
And that causes like undifferentiated cells to exit irreversibly to commit to being differentiated, right?
mhm.
So you could say this is proportional to differentiation. And then they say myode 1 contains a C-terminal nuclear localization sequence, and then they give us a bunch of these single letter abbreviations for amino acids. Whenever you see these, just like look for the ones that'll be charged. Like lysine will be positive one, arginine positive one, um, uh,
There's glutamate and
acid negative one. So okay, plus one, plus one, minus one, and then there's D
mhm.
negative one. So let's see, N D A F E I T K R C. Seems like it might be neutral. Anyway, promotes its translocation to the nucleus. So I'm gonna say like moves to the nucleus. Okay. You can continue.
Okay. Researchers simulated an in vitro biomechanical stimulus environment by using a cyclic tension strain cell culture chamber and investigated the effect of cyclic tensile strain on myogenic differentiation by measuring the levels of myode in the presence and absence of TNF-A. The TNF-A is a pro-inflammatory cytokine that, upon binding to its receptor, TNRF1, induces the expression of nitric oxide synthase, and no production.
All right. So we have some more stuff to do proportionalities for.
Hmm.
I don't even care what these are. Let's just the alpha looks like the proportionality symbol, so just saying that. Okay. Causes TNFR1 causes NOS causes NO production. I'm gonna put it like this to indicate like, concentration of that of the nitrous oxide. All right.
Um, Cultured myoblast cells were subjected to CTS, and the Myod1 mRNA levels were measured in the presence or absence of recombinant human TNF alpha and NG nitroarginine methyl ester, a competitive inhibitor of NOS activities. And these are the results here.
So I'll say NOS inversely proportional to L name.
Hmm.
And CTS probably causes something with Myod1 to happen. But okay, let's look at this chart. So, um, so yeah, these can seem very like overwhelming at first, but just kind of do it in a slow, systematic way. So I would just look at, okay, first bar and y-axis is Myod1 mRNA levels. So we're essentially looking at the expression of Myod1. And yeah, control is at 100%. When you don't have TNF-alpha, you don't have L-name, you don't have CTS. And then let's see, when you have CTS only, it increases Myod1. So I'm going to put CTS here, proportional to Myod1, right? And we can kind of think of CTS as like, exercise.
Yeah.
Okay. And then when we don't have CTS and we still don't have L name, but we have TNF alpha, we have less myode 1. So I'm going to say TNF alpha inversely proportional to myode 1. Okay. Then next, CTS and TNF alpha. So now the question is like, do we compare, what do we compare this to, right? Because we have two things happening. So I would compare them with either one of these on their own. So for instance, when you just have CTS over here, we have more myode 1. And when you just have TNF alpha, you have less myode 1. So when you have both of them, it seems like they just do what they do. So I guess TNF alpha makes it go down a bit compared to like here where it's just so okay, let's do this. TNF alpha causes it to go down, CTS causes it to go up. So you end up
Hmm.
with something in between just CTS where it goes up like really high, like 140, and between that and TNF alpha where it goes down to 40. So it's just somewhere in the middle. Okay. And then when we have TNF alpha and then L name, so TNF alpha is supposed to bring it way down,
Yeah.
but when you add L name, it brings it up a little bit. And we know from what we wrote here, TNF alpha makes NOS, but L name like like inhibits NOS, right?
Yeah.
So it would make sense that L name would inhibit the effect of TNF alpha, um, making it higher than over here when you just have TNF alpha, right?
Yes.
Okay. So yeah, so these proportionalities can really, really help out. Um, and yeah, and then we can try it over here. So let's see. So which compound is predicted to mimic the effect of exercise? So that's the CTS
Mm-hmm.
in undifferentiated muscle cells in vivo. So okay. Um so Myod1 specific microRNA. So that's kind of like so unfortunately like that is something you'll have to kind of know, but typically microRNAs are used to inhibit stuff.
Okay.
So, um, so well, okay, so we know that it should make more Myod1. So if Myod1 specific microRNA inhibits Myod1, that's not what we want, right?
Right.
Uh,
Um, I didn't think that it was NOS either.
an activator of NOS expression. So we know that the TNF-alpha is inverse to myode, and the TNF-alpha does the NOS. So myode. So we know that CTS, aka exercise, should cause more myode, but we know that
Yeah.
NOS is inversely proportional to myode, right?
Mm-hmm.
Yeah,
So
less
yeah.
NOS, not more. So this is out. Uh,
Yeah.
a repressor of CDKN1. Well, we see myode 1 is proportional to CDK and 1A. So
Yeah.
repressor wouldn't work. And an antagonist to TNF alpha, we see TNF alpha and myode 1 are inversely proportional. So if you got rid of TNF alpha, you'd get more myode.
Okay. Yeah, that makes more sense. I didn't know what you're right, I didn't know what that was. That's why I just picked this one.
Yeah, yeah. Yeah, because otherwise that one would seem like it says more myode 1, and that would make sense. But yeah, it's kind of annoying.
Okay.
But yeah, just like
Wait,
proportionality stuff is really, really is going to be really helpful.
gotcha. And then I thought like because it's a protein, no? It says,
Hmm.
well, I thought that you would use Western blotting to investigate the transcriptional regulation of
Ah,
CDK.
so if we're looking at so it is a protein, but the regulation of it on a transcriptional level would be before it's translated, right?
Okay, yeah, that makes sense.
So it can't be, so yeah. So won't be these guys, and it'll be something with mRNA,
It's
snowdrop.
not B. Yeah, Snowdrop. I definitely have that in my mind all the time. Yeah, it's
Yeah.
definitely not B. Okay.
Okay.
Okay. Um, damn. Okay, I didn't see that. I didn't notice that transcriptional regulation thing there, but good to know. It's just a practice test.
Hmm.
Um, I thought it was native page because they kept the proteins in their like native state.
Hmm.
Um, so I guess it's B then, just gel filtration chromatography. What does that mean? Okay. Okay. I don't know.
That was weird.
What?
So I figured that, well, I guess, all right, that's just weird, but okay.
Hmm.
Oh my God, it's not even. Oh, that's annoying. Okay. Apparently,
Wait,
yeah, because I was like, we were both thinking something like this, right? But that like gels like this, they need an electric field, otherwise they wouldn't move. But apparently, gel filtration chromatography is the same thing as size exclusion chromatography.
okay.
I was I was like, what? But yeah, I just looked it up. It's literally it says gel filtration chromatography, also known as size exclusion chromatography. And that's when you separate it based on chart. I'm sorry, on size.
Okay. They are so tricky over at the AAMC.
Yeah.
They don't want to give away points.
Yeah. Yeah, that one was bad.
Okay. Good to know.
Mhm.
Um, this. Oh, wait, no, this. I didn't think I got this one wrong. Actually, okay. Okay. Uh, I would say that because at first I thought it would be A, just because, like, I don't know. Isn't that what you reabsorb water
Yeah.
for?
Yeah, I think this is one of those um annoying ones where you just have to I guess know or learn from this that most of this is through the skin and lungs, but yeah, I remember this one and I remember like it wasn't something that I could like really logically obtain from the passage.
Okay, so this is just like a known fact that like instead of the like after the water is filtered, it's not going to be blood first. It's going to be excreted first.
Yeah, so I think all right, let's see. So drink about 1 liter of water on average and excreted about 400 milliliters. So there's 600 milliliters, which you would think would be stored as blood, of course, but apparently I think it is mostly gone through the skin and the lungs. I don't know. They probably explain it better.
Okay, I'll flag her.
Hmm.
And okay, and
Yes.
it's
Mhm.
yeah. I figured it was either B or D. Like when I was taking the test, and I put D because
Hmm.
it just said that. Well, it's the usual response to dehydration. I thought they wanted for us to like refer to something in the passage, I guess, but
Hmm.
conserving water makes the most sense.
Hmm.
Like, I wonder what they say. Oh, okay. I see. Okay. Mm, this one, I thought it was talking about like it's innate in immune cells, but actually, now that I think about it, hold on. If it's in the nasal, maybe it is this.
Yeah, that'll be the immune cells of the brain,
Oh, okay. Never mind.
blood-brain
So,
barrier. Yeah, although colonization of the nasopharynx is usually symptomatic, in some cases, bacteria enter the blood, migrate to the brain, cross the blood-brain barrier. then that can cause meningitis.
Okay. All right, that makes sense.
Mhm.
Yes. Okay. All right. Isn't that gap junctions?
So gap junctions are like when you have the cells nearby that are connected like that, like have a continuous like cytoplasm.
Mm-hmm.
Um, but
Right.
the blood, yeah, that's like the watertight um kind of thing that prevents,
The drugs.
yep, and like
Okay.
all things, yeah.
I had this one down. This is the endomembrane system, right? In the no, 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 no. I'm bugging. No, that's not it. That's it's the the packaging system, like when the cells do the things.
Signal sequence and stuff.
Yeah, I thought I had it at that, but I just gotta look at my notes for this one then.
Hmm.
I thought it started at the Golgi complex and then to the rough ER, but maybe it's the rough ER and then the Golgi.
Yep. So it's
Okay.
like when a protein is being translated, if there's a specific sequence of amino acids called a signal sequence, it would pause the translation, and then the ribosomes will move to the endoplasmic reticulum
Hmm.
and resume the protein translation, but it resumes it like inside of the endoplasmic reticulum. Um, and then it can go to the Golgi, and then it can be going through like a vesicle to either, someplace at the end of the cell, like in the cell membrane, or something like closer, like the nucleus. But
Okay.
be like, yeah, if you look up like signal sequence, that's what this would be.
Okay, signal sequence.
Mhm.
I have that in my notes. I don't know where I got that one from then. I must have just seen someone say it and was like, okay, that's what this is. Um, and honestly, this one kind of has me a little confused. Like it says white eyes are recessive to red eyes, and yellow bodies are recessive to dark bodies, and they're both on the X chromosome. So if a genotype of a red-eyed and a dark body female are XX could be determined by mating it with a male that has which of the following? Oh, it says could be determined. Hmm, that's crazy. Okay, that makes more sense. Because I thought like I don't know. I think I was just assuming that they were trying to like have that genotype happen again. So I was like, oh, okay, like red eyes, dark body. Um, but if they're trying to figure out what the genotype is, then I guess it's
Hmm.
um
Yeah, because if red is dominant and dark bodied is dominant, and let's just say I'll just use A's, like we don't know if this female is homozygous dominant or heterozygous dominant, right?
yeah. So she would have it would be like a male with recessive genes then. Oh, I could have got this one right. That's okay. But yeah. Um, I
Yeah,
don't know.
I think you're good. I think that this week, just like for the most part, like take it easy. Do like some Anki review, do like passive review and stuff, but like really dial in like your sleep and all of that stuff.
Okay. Should I keep doing like more practice questions just to like get used to, well, continue to get used to like the AAMC logic?
Uh, Yeah, but not like yeah, not like an extraordinary amount. Let's and your test is on Friday or Saturday?
On Saturday.
Okay. So let's I would say like by Wednesday, um or I guess Thursday because we're gonna meet then. But like Thursday will be like the last day where you do like practice questions for like, Friday. Um yeah, so I guess I'll say like make sure you dial in your sleep, your um everything else like that. Um and yeah, maybe you could do some questions but not like an extreme amount and probably like around Wednesday or Thursday will be like the last like questions that you do. After that, just do like kind of passive review. Look through like the quick sheets. Look through like a lot the PDFs in the PDF folder. Um, think about like some of that stuff that you can like kind of brain dump like on the scrap paper that you have in like the first 15 minutes. But yeah, I would say don't go too crazy.
Okay, yeah, I think I'll just do practice questions. I don't know. I feel like I'm just nervous. Like I really wanted
Mhm.
to improve a little bit more. Like, I don't know, see if I could get at least like a 505 or something like that.
Yeah, I think
Um,
you're on the road to that. Um, but it's very, very stressful and there's a lot of things going on and all that stuff. So I think the 500 is still really, really good. It's probably like a lower bound. And you're probably going to get someplace, 502, like above 502. But just, yeah, just dial in your sleep and your nerves and all of that stuff.
okay, yeah. Well,
Mhm.
thank you. Um, and I guess I'll see you on Thursday.
Yep. All right. Oh, and then let me put in the it should be 160 for today.
Okay,
All right. Take care.
you too. Bye.